SUMO-binding entities make it possible to isolate endogenous SUMO-related proteins in vivo. This is rather challenging due to the presence of active SUMO-specific protease and the low fractions of SUMO-related proteins in vivo.
The isolation of the SUMO-related proteome by using SUMO-binding entities is advantageous as it offers an increase in the overall affinity for SUMO substrates. This is due to the fact that SUBs are recombinant proteins that comprise tandem repeats of SIMs, thereby recognizing specifically SUMO molecules on modified proteins.
The use of SUMO-binding entities for the isolation and characterization of the SUMO-related proteome in liver cancer is a fast and sensitive method. It provides vast information on the rather unknown role of the SUMO-elation pathway in liver cancer and a potential therapeutic approach.
SUMO-binding entities can be used to isolate and characterize the SUMO-related proteome in other tissues besides the liver, both from human specimens and animal models, as well as in several cellular models.
Although this technique is easy to perform, care should be taken when analyzing several samples at the same time, as confusion could occur due to the several steps involved. Therefore, we recommend careful labeling of all the tissues before initiating the experiment.
The visual demonstration of the use of SUMO-binding entities facilitates the reproduction of this protocol, especially due to the difficulties of handling the bits and the eventual loss of material during the protocol.